Okay, we're back on the clock. We'll now move on to discussing genotoxicity, but first we'll look at what we mean by some relevant terms. In the public mind, terms like toxin and poison are often seen as synonyms. This is not strictly speaking the case, although there is some overlap. Poisons refers to a relatively short list of substances that may, in some circumstances, be sold to the public. For example, rodent poisons. Toxin is a designation that is applied to a much wider range of substances that are used in industrial and other employment-based circumstances. Click on this image, we'll take you to an article from Corona if you're interested. Toxins and a range of other substances that may present risks to human health include microorganisms pathogenic in humans, and more recently nanomaterials, are controlled by the COSH regulations, the Control of Substances Hazardous Health regulations, which you will be familiar with from the information we supply to you when you're doing chemistry type labs, or biology labs for that matter. Again, there's a link there to HSE's COSH website. Many of you are familiar with at least some of the older symbols, possibly from school or college labs. These have been replaced by a new set, highlighted here in red, including some new hazards. So you might want to pause the video and try to identify the hazards the new symbols are associated with. Okay, I'll assume you've done that. Bottom left of the image is the new longer term serious health hazard symbols. This may be relevant to some nanomaterials. The caution symbol has replaced the previous harmful symbol, and there's a new symbol for gases under pressure. Again, click on the image for a link to HSE. All food applications use cleaning materials such as bleaches and disinfectants that may present hazards to health. Specific industries such as baking may have their own issues. In that case, for example, the problems arise from flowers and other dusts, which may be explosive among other things, as well as presenting respiratory type hazards. Again, click on the image for more details if you're interested, or see the notes in the video notes. Two other definitions. When we talk about toxicity, we need to distinguish between substances that can cause death quickly, said to be acute toxins, and those that produce adverse effects across the lifespan. Acute toxicity is assessed using LD50 data, this being the amount of substances required to cause death in 50% of a group of test animals. Leaving aside other issues, acute toxicity is relatively easy to assess, whereas chronic toxicity, including genotoxicity, is much harder to evaluate. Okay, we're now looking at genotoxicity and potential issues with nanoscale titanium dioxide. Genotoxicity refers to the ability of harmful substances to damage genetic information in cells. Being exposed to chemical and biological agents can result in genomic instabilities and/or epigenetic alterations, which may translate into a variety of diseases, cancer included. Epigenetic effects are those that may be translated across generations. For those who are doing nutrition, we'll do more on that in other modules. This review article discusses the various methods that have been used to assess the potential genotoxicity of TiO2. The details of the methods are well outside the scope of this module, but you should be aware of the general approaches taken. And the article, again, is included in the notes. We'll notice there's in vivo and in vitro methods, as you would expect. So proposed mechanisms, one of the key ones is oxidative stress. We've discussed before in the first year, oxidative stress is associated with an excess of free radicals in tissues. See these first year lectures for reminders of the background. Oxidative stress can damage cells, proteins and DNA, which can contribute to aging and longer term health effects. Titanium dioxide nanoparticles have significantly higher surface area than bulk particles. This is associated with a high rate of free radical production. Assays suggest that smaller titanium dioxide particles are more genotoxic than the larger particles, for reasons we discussed earlier. Some background here really, I guess. Base excision repair, BER pathway, and nucleotide excision repair, NER pathways, are two mechanisms which are involved in these processes. See the paper if you're interested, but this is somewhere outside the scope of the food chemistry module. The basic concepts may be examinable, but certainly not the details involved in this paper. This review concludes that although many tests have detected genotoxic effects, there are clear inconsistencies in the reported results. These inconsistencies may have arisen from physical differences in the test materials and different methodologies. In general, in vitro systems generated more positive results than in vivo systems. This is often the case in a wide range of studies in a whole different set of areas. In vitro are easier to control and run than in vivo systems. This paper covers a similar background to Chen et al., but includes a short section on proposed directions for further study. Again, the details of background only. The point here is to demonstrate the considerable limitations in our understanding of these issues. The OECD is an organisation that assists countries in harmonising guidelines for the testings of chemicals and good laboratory practice in order to assure high quality and reliable data for countries and industries to fully benefit from the OECD Agreement on Mutual Acceptance of Data, acronym MAD, and to avoid duplicative testing. So some interesting background for anyone who might be thinking about a career in that sort of area. Okay, that's all I want to say about the evidence, which is, as we said, developing, shall we say, but some exam-related exercises for you to think about. This is my map of lecture content in the order in which I wrote the various topics. The map is condensed down to the main headings. The XMAN file, which includes the notes, and you can look at the fully expanded version. You may think another order is more logical. There's just a little zoom in on nanomaterials. An exercise, exam related. As we mentioned, we are going back to pen and paper exams, unseen pen and paper exams. Mind maps can be very useful to help you remember the main points of a detailed answer. So sketch your own mind map of the lecture content, not just this one, and rough is fine. There's no need to use the software, and I'd strongly recommend that you don't. Make sure you draw the main points from memory. And here, again, a logical approach from your point of view will help. So here's a possible exam question covering many different issues. It's possible that this 15-mark question could be broken out into smaller questions, but it's a good idea to be able to stand back and look at the wider picture. For a moment, we'll consider how to approach the overall question. It's often a good idea to briefly summarise the issue in plain language. Generally speaking, when a discussion is a complex issue in depth, you need a general overview to start with, and then you move down to detailed examples. Always go from the general to the specific. That's true of all academic writing. Also, when looking at an issue, you should define your terms first. In this case, we could look at the industrial applications of TiO2, maybe briefly mention non-food applications, but the bulk of your answers should be food related. We'll look at this answer shortly as an example. The section health issues would have been placed before the scope section, but both cases ensure you discuss these issues in the context of TiO2. So here's an 8-mark question. So in terms of a traditional exam, you should not be spending more than 10 minutes on it. We'll discuss this in tutorials during normal teaching sessions. You will be expected to develop your own approach to understanding these issues. Again, here the links. I'll post these in the YouTube video description, but obviously you can get them from the notes and presentations. Right, that's all, folks. Thanks for listening.